In this video, we will discuss about this antibody and bispecific T cell engagers. So we will discuss the structures about these two things. And this bispecific T cell engagers or BiTE is dependent on this antibody. So we will start with this antibody structure. So antibodies has two different regions, that is FAB and FC. FAB is fragment antigen binding region, and FC is fragment crystallizable region. This fragment antigen binding region contains this variable heavy chain, variable light chain, and this constant heavy chain, and it has two different sides of this structures, that is variable heavy chain and light chain is also on the other side, and this constant heavy chain and the constant light chain, and these two are connected by this disulfide bond. And this fragment crystallizable region contains this constant heavy chain 2 and constant heavy chain 3 region, and this variable heavy chain and variable light region is the one that has the specific binding affinity against a target for which this antibody has been raised. So this is the general structure of antibody, how it is been used to make this bispecific T cell engager. So in the bispecific T cell engager, what they did is they took the variable heavy chain and light chain region of one monoclonal antibodies and from another monoclonal antibodies and joined together to form a BiTE antibodies. The linker that they used is the serine glycine linkage. It can be seen here between heavy chain and light chain of these two, and also this light chain and heavy chain of the other monoclonal antibodies. And what are these two monoclonal antibodies? First one is to target T cells, that is anti-CD3 monoclonal antibodies, and second one is related to this tumor associated antigen monoclonal antibodies that can target the tumor antigen. And once they are linked together using serine glycine linkage, then they will form a polypeptide, and that polypeptides will form like this heavy chain connect to the light chain connect to the heavy chain, and this light chain. And the role of this bispecific T cell engager is to engage the T cells with the tumor cells, that is one side will bind to the T cells and the other region will bind to the tumor associated antigen. So it will bring together the immune cells and the tumor cells, so that the immune efficacies against the tumor cells can be enhanced. And this has shown prominent results in the animal mouse models, and so this could be one of the therapeutic approach that can be used to diagnose tumors, especially the solid tumors, because solid tumors, these solid tumors generally have these characteristics of the tumor microenvironment that creates tumor evasions, that creates immune evasions, not tumor evasion, that creates immune evasion and helps in the tumor proliferations. So if they use this BiTE, then this might suppress the tumor evasions and increase this tumor suppression. So this BiTE antibodies is a prominent prominent approach that could be designed in a way to target the solid tumors. However, it is not related to the not restricted to the solid tumors. It can also be used for the other cancers such as blood cancer, myeloma cancers, that can also be used in this BiTE antibodies can also be used in this that treatment. However, in the non-solid tumor cancers, the other methods are also very effective, and one of the prominent method that is currently being under clinical trial is in vivo CAR T cell generations. So that will eliminate the need of the lab scale modifications, that takes lots of time. So this is just how this antibody and bispecific antibody structures looks like and how they are designed. So if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.